Today I'm actually going to tell you, for the sake of time, about just one of three parts of the study that I'm doing under the auspices of funding from the Children's Discovery Institute. And we're testing a hypothesis, as Dr. Dunauer mentioned, about how to devise a new treatment for retinoblastoma. So I think it's good to start with giving you an idea of what retinoblastoma is. Retinoblastoma, as was briefly mentioned, is the most common cancer of the eye in children. It tends to affect fairly young children under the age of 5. There's about one case amongst every 15,000 children, and there are as many as 3,000 deaths a year, but most of those are in the developing world. We're getting progressively better at treating this disease in developed countries. This cancer arises in the developing retina, which is the light-sensitive portion of the eye. It's a thin layer of tissue at the back of the eye, and I'm going to tell you more about that in a minute. When a patient develops this disease, the treatment often involves chemotherapy or radiation to the eye, and unfortunately, in many cases, actually, it necessitates surgical removal of the eye because the cancer cannot otherwise be treated. And so, it turns out that about a third of patients develop these tumors in both of their eyes. So then obviously you run into a real problem, and that you'd like to avoid, if at all possible, having to remove the eye, because otherwise you make the patient permanently blind. So that's why we are trying to develop novel approaches to treating these cancers that don't require surgical removal of the eye. And so we'll start by showing you a picture of a young child. This is a 3-year-old girl that has a retinoblastoma. And you'll notice if you look closely that there's a subtle difference in her two eyes. Okay? So everyone is used to this phenomenon. When you take a picture with flash camera, often you get the red eye effect. Well, you can see that she has that red eye reflex in her left eye, but not in the right eye. In the right eye, she has a white. And so there's a difference between the two. And this is often the way retinoblastoma is first detected. So it's a parent or a relative that will first detect it, say when a picture is taken with a flash at a birthday party or something. And they'll notice that there's a difference between the two eyes. And what's going on inside the eye is the following. So normally, in a normal eye, when a camera flashes, the white light will go into the eye, it will literally bounce or reflect off the back of the eye and come back out, but it'll come back as red light. And the reason it does is because the back of the eye is covered with blood vessels that have lots of red blood cells in them. So that essentially causes a red reflection from the eye, and that's where you get this red eye effect. That's a normal eye, so that's her left eye here. However, in a patient with retinoblastoma, you have an abnormal mass of cells that develop in the back of the eye, and that will be white or yellowish colored. And when the flash of the camera comes in, it will bounce off of that instead, and it will come back white. And so that will give you an appearance like this. And so this is very frequently the way one of these tumors is first detected. So in order to understand how this tumor comes about, I need to explain just a little bit about how the human retina works and allows us to see, and then tell you briefly about how the retina develops. So normally during vision, light comes in through the pupil, passes through the eye, and has to reach the retina, which is a thin layer of tissue here lining the back of the eye. So it's the light-sensitive part of the eye. And so then the light comes in, passes through the retina, and this is an H&E stage section of human retina, and it comes in and it impinges on the photoreceptor cells. And these are the cell types that are actually light-sensitive. And so they can do a very remarkable process in that they can convert a light signal into an electrical signal. And neurons communicate by electrical signals. And so then that electrical signal is essentially processed by the retina and passed back to the brain as visual information, essentially reconstructs the image that we see. Okay. So in a nutshell, that's how the human retina works, and that's how it allows us to see normally. Now, the two cell types that are involved in light sensation are called rods and cones, and many of you have probably heard of those. The rod photoreceptor cell is responsible for vision at night, so it's responsible for dim light vision, and the cone is responsible for vision during the daytime, so bright light vision and color vision as well. So the process of retinal development, how the retina arises during the development of a human being, goes as follows. You start out with an embryonic retina, and it's basically a homogeneous layer of cells that are all more or less alike. These are progenitor cells. And they undergo a process of growth and differentiation to ultimately give rise to an adult retina. Now, development of the retina or any other organ in the body doesn't, or growth doesn't occur by a simple process of enlargement. So, for example, a fetus's cells or the cells of a young child are about the same size as the cells in an adult's body. So how growth occurs is by a process of proliferation rather than growth of individual cells. And by proliferation, I mean that a cell will undergo a process of division that will give rise to 2 cells. And then those 2 cells will each give rise to 2 cells and so on. So they'll continue to divide and proliferate in that way. And so that's how the process of development occurs. And then once they've proliferated a lot, then these cells eventually undergo differentiation. And by differentiation, we mean that they go from being an immature state to a fully differentiated mature state and have their mature function. Okay? So then, in a nutshell, the process of retinal development means going from a small number of progenitor cells through proliferation and differentiation to give rise to the 7 mature cell types of the retina. This can be pictured in a slightly different way. And I think it's important for you to understand this in order to get an insight into what we're trying to do in our research. Let's conceive of retinal development as a ball that's rolling down a landscape like this. And so this is an immature progenitor cell, and it's proliferating. It's undergoing a process of cell division and giving rise to more and more cells like this as it rolls down this landscape. And once it finally reaches the bottom in any one of these valleys, that is essentially the differentiated state, where it stops proliferating and then reaches its mature form. Okay? Now, what happens when a child develops retinoblastoma is that this process of rolling down the hill and differentiating is interrupted. It's disrupted. And the reason it's disrupted is because these children have genetic mutations in a gene called RB1, or retinoblastoma 1. Okay? And so when the function of RB1 is lost in a single cell in the developing retina of this child, it basically establishes a roadblock to differentiation. So that cell can no longer keep rolling down here to differentiate as a rod or cone. Okay? So as I said, all these intermediate stages of these immature progenitor cells are growing and dividing all the time. And so what happens is that cell continues to grow and divide, and the process carries on and on until you have formed a disorganized mass of cells, which represents the retinoblastoma itself. That's the tumor. Okay? So then the idea is that there is an arrest or a disruption of the terminal differentiation process. The cell gets hung out in this state where it continues to proliferate out normally. So just taking a step back then, this is where we're at. And what we wanted to figure out, and still would like to figure out, is what exactly is the nature of this roadblock? Why can the cell not continue to differentiate? And one clue that we have recently discovered is that there is another gene that is controlled by RB1 that is required for the differentiation of rod photoreceptors. That gene is called NRL. And so basically, RB1 normally is required to turn on NRL. And NRL is required for this last step of differentiation. So the point is then that if you lose RB1, you don't turn on NRL, and you can no longer complete that differentiation step. Okay? This is the hypothesis. And so the idea would be for novel therapy that if there was a way to turn on NRL, even in the absence of RB1, that is, to turn on the NRL in this abnormal tumor cell here, that you might be able to force that cell to differentiate, force it to assume a mature state, to stop proliferating, and either become a normal photoreceptor or simply be a cell that's hemoglobin divided.